Hi all. In the last tutorial, we have discussed about the history of microbiology and the contributions of some eminent scientists. In this tutorial, we will discuss more about bacteria. Bacteria are cosmopolitan, and the term bacterial was described by Ehrenberg. And the study of bacteria, you all know that it is termed as bacteriology. And the smallest bacteria is Dialister pneumonius is citus, and largest bacteria is Epilobium fischelsoni. Now you can know with the quiz on bacteria. The bacterium Escherichia coli is found mainly in dash. You can know yourself. The gram-positive bacteria can be identified by the gram staining because of the recent dash. You all know gram staining, and it was described by Hans Christian Gram in 1884, and he classified the bacteria based on the color compounds, fixation of color compounds, and the bacterial cells found in cubical packets. The shape of Vibrio bacteria is dash. The classification based on shape you have to know. Bacteria that forms an endosymbiotic nitrogen fixing association with roots of legumes that helps in nitrogen fixation. In our last tutorial, we discussed about the contribution of Vinogradsky, in which he described how the bacteria involved in nature. From that, the kingdom represents bacteria. These you can know by yourself. And Anton von Leeuwenhoek in 1676, he was actually a lens maker who first observed bacteria, and he was considered as the pioneer in the invention of microscope. The term bacteria in Greek it's termed as small stuff. It was used in 1838. Robert Koch and Louis Pasteur were the first to discover that bacteria cause many diseases. The bacteria in the mid 19th century, in the last tutorial, we discussed about the contribution of Robert Koch and Louis Pasteur in bacteriology, and they were the pioneer who discussed that various diseases are associated with the bacteria. First antibiotic invented by Alexander Fleming was penicillin. An antibiotic used to treat bacterial disease was made by Paul Ehrlich in 1910, and it was used to treat syphilis. And bacteria, coming to the general characteristics of bacteria. Bacteria are prokaryotic organisms and comes under the kingdom Unira. Prokaryotic. You know that there are kingdom containing the prokaryote and eukaryote in the zoology classes, and they do not have cell-defined organelles like mitochondria, Golgi bodies, endoplasmic reticulum, etc. These are the cell components in the eukaryotic species. And in the bacterium, they do not have such as well-defined organelles. They are microscopic and they are unicellular. They may occur singly or in single small groups to form colonies. They are multiplied in colonies. They possess rigid cell wall. Cell wall is made up of peptidoglycan, purines, and lipopolysaccharides. Absence of a well-defined nucleus. You cannot see the bacteria under microscope. When we observe, we cannot see a well-defined nucleus. DNA is not enclosed in a nuclear membrane. The cell component and the nuclear components are DNA, and these DNA are not enclosed in a nuclear membrane. It is simply displaced in a nucleus. Ribosomes are scattered in the cytoplasmic bacterium and are of 70 S type. The ribosomes and are scattered in this. You can see the cytoplasm is the ribosomes are scattered throughout, and they are of 70 S type. 70 S type means if you centrifuge, you get the molecular weight as 70 S. The plasma membrane is invaginated to form mesosomes. Most of the bacteria are heterotrophic, autotrophic, Chemotrophic, these are all the level of food web. And you know that autotrophies are organisms that can prepare their own food, and heterotrophic means they depend on other substance for as their food. Some bacteria are autotrophic also, possess bacteriochlorophyll, that is, which helps in photosynthesis. Motile bacteria possess one or more flagellae. These are the characteristics of bacteria we are explaining. And flagellae is used as a hair-like appendages protruded from outside from the cell body, and it is one or more flagella can turn this amphitrophs, periotrophs, monotroph, the monotrophic, and these are classification of flagellae, and we will discuss in the another tutorial about the classification of flagellae. And in the bacteria that are motile, they can move with the help of this flagellae, and this flagellae amphitroph means two flagellae and on the either side, and peritrophic means the flagellae are surrounded around the cell wall, and this helps bacteria to move. The common method of multiplication is binary fission. How the bacteria reproduce? It is by binary fission, that is, two cells disintegrated into a single cell divided into two by binary fission. True sexual reproduction is lacking. There is no sexual reproduction we can define in bacteria, but genetic recombination occurs by conjugation, transformation, and transduction. And we will end up this tutorial with the uses of the bacteria in many industry, and many applications are therefore. In fact, there are many diseases associated with the bacteria, but there are uses of bacteria. And in dairy industry that is lactobacillus bacterium, that is used for fermentation of lactose sugar to form lactic acid, that is in curd, and preparation of fermented foods such as cheese, pickles, soy sauce, sauerkraut, vinegar, wine, and yogurt. These are uses of bacteria in the dairy industry. And in food industry, the Bacillus megatherium bacterium is used in the flavoring of tea and tobacco. Acetobacter aceti is used in preparation of vinegar from alcohol. And in industrial uses, we had discussed about the dairy, then we discussed about the food industry, and now we closely observe what are the applications in the industries. Clostridium acetobutylicum is able to produce acetone from acetic acid as well as butanol from butyric acid. And in biogas plant, methanobacterium is used for the production of methane. And bacteria are useful in the fiber ratting, in which the fibers of jute, hemp, and flax are prepared. And the Clostridium butyricum is used in the process of these bacteria hydrolyze the middle lamellae of these plant fibers. And microbial mining, which is the bacteria and other organisms are cultured and contained and then used to bring these processes, example copper extraction, iron extraction, which involves bacteria called ferrooxidants. And the bacteria are natural scavengers, and they can able to decay huge amount of animal and human waste. And using biotechnology techniques, bacteria can also be bioengineered for the production of therapeutic proteins such as insulin, growth factors, or antibiotics. Insulin is used for the treatment of sugar, diabetes. It means diabetes. Some bacteria living in the gut of cattle, horses, and other herbivores secrete cellulase, an enzyme that helps in the digestion of the cellulose content of the plant cell wall. That will be helpful for the digestion, proper digestion. And cellulose is the major source of energy for these animals, herbivorous animals. And generally, plant cells contain cellulose, and the bacteria present in the stomach of cattle will help them to digestion of cellulose. And E. coli that live in human large intestine synthesize vitamin B and release it for human use. And similarly, Clostridium botulinum is used for commercial preparation of riboflavin and vitamin B. Bacillus thuringiensis is also called BT, a gram-positive soil-delving bacterium, is used for pest control and produce BT crops, that is biotechnology applied crops. Bioremediation techniques such as oil zapper use bacteria. There are many applications for bacteria, and many antibiotics are used from bacteria. Some of them are bacteriocin, polymyxin B, streptomycin, erythromycin, neomycin B, chloramphenicol, etc. And an interesting factor I noted about the bacteria is that you can experience the feelings of a pleasant smell after a shower, first rain. I mean, shower means rain, and the pleasant smell of the earth after the first shower, that is earthy odor, is caused by the production of a series of streptomycin metabolites called geosmins, and these substances are secretophenol compounds and unsaturated compound of carbon, oxygen, and hydrogen. The geosmins first discovered has the chemical name trans-1 10-methyl trans-9 decolol. However, other volatile products produced by certain species of Streptomyces may also be responsible for the characteristic smell. So, the pleasant smell after the first shower is it's an interesting factor about the bacterium Streptomyces species. And the last in this tutorial is the biodigester toilet. That is in decomposition mechanism toilet system by means of which the sludge, human waste, the fecal matter is decomposed to bits in the digester tank using a specific high graded bacteria, further converting them into methane and water discharged further to the desired surface. The biodigester toilet is total maintenance free system and does not require any sewage system. And that's all about the characteristics and application or uses of the bacteria. Thank you.